Well, Dembski offers his readers a calculation showing that the flagellum could not have possibly have evolved. Significantly, he begins that calculation by linking his arguments to those of Behe, Michael Behe. We're talking about bacteria and the flagellum, the little squiggly little tails all over this thing, could not have possibly evolved, because he thinks it had to be created because the design was so significant that the thing could not have evolved, and I agree. But he writes, Dembski writes, I want therefore in this section to show how irreducible complexity is a special case of specified complexity, and in particular I want to sketch how one calculates the relevant probabilities needed to eliminate chance and infer design for such systems. So Dembski then tells us that an irreducibly complex system like the flagellum is a discrete combinatorial object. What this means is, as he explains, is that the probability of assembling such an object can be calculated by determining the probabilities that each of its components might have originated by chance. Brilliant idea! And that they might have been localized in the same region of the cell. Brilliant idea! That they would be assembled in precisely the right order. Third brilliant idea. Yes, how do these things happen? Even if by chance these things have to happen just right so that the bacteria will survive. Dembski refers to these three probabilities as Porig, Plocal, and Pconfig. Now, he's scientifically examined it. This writer who wrote these words here, he's never did anything scientific in his article. He just pounded on things, but didn't show proof. And he regards each of them as separate and independent. This approach overlooks the fact that the last two probabilities are actually contained within the first. Localization and self-assembly of complex protein structures in prokaryotic cells are properties generally determined by signals built into the primary structures of the proteins themselves. Yes, okay. How are those signals built into the primary structures of the proteins? Did they happen by chance too? You're making it worse. Well, okay. The same is likely true for the amino acid sequence of the 30 or so protein components of the flagellum and the approximately 20 proteins involved in the flagellum's assembly. How does that happen? It's just mind-boggling to think that this all just fell into chance. Therefore, if one gets the sequences of all the proteins right, localization and assembly will take care of themselves. I say actually not. Signals might be sent out while the components have not yet evolved by chance, in which case nothing would be accomplished yet, and the configuration activity might be working, but with no component in the proper position to make installation effective. So it is evident that all three have to occur at once. There you go. Plus, who invented the signals? To the ID enthusiast, however, he goes on to say in the dark fonts, this is a point of little concern. According to Dembski, evolution could still not construct the 30 proteins needed for the flagellum. That's right. His reason is that the probability of their assembly falls below what he terms the universal probability bound. Yeah, like if it's older than the universe. According to Dembski, the probability bound is a sensible allowance for the fact that the highly improbable events do occur from time to time in nature. To allow for such events, he agrees that given enough time, any event with a probability larger than 10 to 150 might well take place. Therefore, if a sequence of events, such as a presumed evolutionary pathway, has a calculated probability less than 10 to 150, we may conclude that the pathway is impossible. The problem with this is none of this has happened. So, to repeat, if the calculated probability is greater than 10 to 150, it's possible, even if unlikely. So, he goes on to say, when Dembski turns his attention to the chances of evolving the 30 proteins of the bacterial flagellum, he makes what he regards as a generous assumption, guessing that each of the proteins of the flagellum have about 300 amino acids. One might calculate that the chances of getting just one such protein to assemble from random evolutionary processes processes would be 20 to 300, since there are 20 amino acids specified by the genetic code. Dembski, however, concedes that proteins need not get the exact amino acid sequence right in order to be functional, so he cuts the odds to just 20 to 30, which he tells his readers is on the order of 10 to 39. Dembski wrote this in 2002. Says the flagellum requires 30 such proteins. He explains that 30 such probabilities will all need to be multiplied to form the origination probability. That would give us an origination probability for the flagellum of 10 to 1170, far below the universal probability bound. The flagellum couldn't have evolved, and now we have the numbers to prove it, right? Suppose, I say, the time frame for this exceeds the proposed age of the universe, just for this one simple life form, never mind mammals and man. So, assuming impossibility. All right, dark font. This author, who is against creationism, an intelligent designer. Assuming impossibility, says, I have no doubt that to the casual reader, a quick glance over the pages of numbers and symbols of Dembski's books is impressive, if not downright intimidating. Nonetheless, the way in which he calculates the probability of an evolutionary origin for the flagellum shows how little biology actually stands behind those numbers. You don't have to have biology, more you have to have math. His computation calculates only the probability of spontaneous random assembly for each of the proteins of the flagellum. Having come up with a probability value on the order of 10 to the 1170, he assures us that he has shown the flagellum to be unevolvable. 10 to the 1170th power. This conclusion, of course, fits comfortably with his view is that the Darwinian mechanism is powerless to produce irreducibly complex systems. However, complex Dembski's analysis, the scientific problem with his calculations is almost too easy to spot. Oh, really? By treating the flagellum as a discrete combinatorial object, he has shown only that it is unlikely that the parts flagellum could assemble spontaneously. I think it's EL. No, it is assemble. Unfortunately for his argument, no scientist has ever proposed that the flagellum or any other complex object evolved that way. Dembski therefore has constructed a classic straw man and blown it away with an irrelevant calculation. By treating the flagellum as a discrete combinatorial object, he has assumed in his calculation that no subset of the 30 or so proteins of the flagellum could have biological activity. As we have already seen, this is wrong. Nearly a third of those proteins are closely related to components of the TTSS, which does indeed have biological activity, a calculation that ignores the fact that has no scientific validity. Well, more importantly, let's see if I got an answer to this. So far, but you have actually observed a bacterium with a locomotion component in which you replace the component with another functioning component that is similar or identical found in another species that is similar to the first bacterium to see if it functions properly, either for locomotion or for other purposes such as producing toxins. No, he hasn't. So he didn't do it in a scientific assembly. So he's saying this could have evolved and that could have evolved, but you haven't saying, well, how would it be if there was a substitution there? You replaced a component with another functioning component that's similar or identical. Haven't done that. That's all his wording up here. Have you found any bacterium without a functioning locomotion component? In order to get the locomotion component, the bacteria has to exist, but it can't exist without the locomotion. If you have not done any of these things, then your conclusions are pure speculation with no real basis for speculating, hence a waste of time. Note that an intelligent designer cannot be ruled out in any case because he could have created such functioning components as well to be received by the life forms he created. But just as he created sources of liquid and food to have it readily available for the life forms he created to utilize, in order to sustain itself, so he could have created such components for the life forms to receive and place them in such a manner, in such quantities, to assist his creation in surviving. So the question is, have you found any of these components floating around in the environments that the bacteria occupy? If not, then don't speculate further. Okay. And none of these things has to evolve over a long time, but be made immediately available for life forms God created, which live upon the earth that he created. That's the point to all this diatribe. I didn't want to keep reading them. It goes on and on and on. It makes the presumption that what he's saying could exist, but never found anything that did exist that he's talking about. So in any case, we need to move on. When anti-evolutionary arguments featuring the bacterial flagellum rose into prominence, beginning with the 1996 publication of Darwin's Black Box by Michael Behe, they were predicated upon the assertion that each of the protein components of the flagellum were crafted in a single act of design to fit the specific purpose of the flagellum. The flagellum was said to be unevolvable, since the entire complex system had to be assembled first in order to produce any selectable biological function. This claim was broadened to include all complex biological systems, and asserted further that science would never find an evolutionary pathway to any of these systems. After all, it hadn't so far, at least according to one of design's principal advocates. Michael Behe says, there is no publication in the scientific literature in prestigious journals, specialty journals, or books that describes how molecular evolution of any real complex biochemical system either did occur or even might have occurred. A note. Then in the article, I respond, you actually do admit to not knowing the details of how anything evolved. And he doesn't bother experimenting to see if maybe it could. Living cells are filled, of course, with complex structures whose detailed evolutionary origins are still not known. And as many critics, he goes on to say, of intelligent design have pointed out, that statement is simply false. Consider just one example, the Krebs cycle, an intricate biochemical pathway consisting of 9 enzymes and a number of cofactors that occupy center stage in the pathways of cellular metabolism. The Krebs cycle is real, complex, and biochemical. Does it also present a problem for evolution? Apparently yes, according to the authors of a 1996 paper in the Journal of Molecular Evolution, who wrote, the Krebs cycle has been frequently quoted as a key problem in the evolution of living cells, hard to explain by Darwin's natural selection. How could natural selection explain the building of a complicated structure in toto, when the intermediate stages have no obvious fitness functionality? Where intelligent design theorists throw up their hands and declare a defeat for evolution, however, these researchers decided to do the hard scientific work of analyzing the components of the cycle and seeing if any of them might have been selected for other biochemical tasks. Again, it's the same thing. Take my battery in the Ford and put it in the Toyota. What they found should be a lesson to anyone who asserts that evolution can only act by direct selection for a final function. In fact, nearly all of the proteins of the complex cycle can serve different biochemical purposes within the cell, making it possible to explain in detail how they evolved. My answer, although you stipulate that a number of the components of the Krebs cycle have been observed to participate in different biochemical purposes within a cell or other cells, this does not make it possible for you to explain in detail how they evolved. There has to be more information which indicates the progression of that evolution stage by stage, failure by failure, success by success, over many years, lots of detail, which most likely require you to be very, very old, millions of billions of years old, because you have to be there to observe. For evolution, by its own self-description, is not straightforward progress, but a series of virtually endless failures interspersed with successes here and there until a huge amount of trials, uncountable years, go by, and you have some constructive results that you have called random processes and natural selection, as well as survival of the fittest. Since both creationism and evolution are faith-based models of how the universe, Earth, came about, because neither is sufficiently observable in how it came about, to be there as each species evolved or did not evolve successfully.